Hello, we shall discuss on the Watson and Crick model of DNA. In 1953, two scientists named Watson and Crick proposed a model to explain the structure of DNA, called as the Watson and Crick model. Now we shall discuss on the features of this Watson and Crick model. According to them, the DNA molecule is formed of two polynucleotide chains. So these are the two polynucleotide chains of the deoxyribose nucleotide. Each of these deoxyribose nucleotide consists of a phosphoric acid attached to the fifth carbon, a pentose sugar, and a nitrogenous base. Now, each of these nucleotide is attached to the adjacent nucleotide from a bond called as the phosphodiester bond. Hence, the deoxyribose sugar, the phosphate group, and the nitrogenous base form the backbone of the polynucleotide chain or the DNA. The two polynucleotide, that is 1 and 2 strands, are arranged anti-parallel. One is running in this direction 3 to 5 direction, and the other is running in this direction 3 to 5, but in opposite direction. So this is the meaning of anti-parallel strand. So these two anti-parallel polynucleotide strands are arranged spirally, forming a staircase type of structure. So this is showing the staircase like structure. So spirally arranged in a form of a staircase like structure. The DNA helix, this DNA helix has a diameter of 20 angstrom or 2 nanometer. That is from here to here. The distance between one nucleotide, one of the nucleotide to the opposite nucleotide is 20 angstrom or 2 nanometer. Now the bases between the two polynucleotide, that is nitrogenous bases, is linked by a hydrogen bonds. Hence pairing is such that in case of we know that adenine always pair with thymine. So the pairing between adenine and thymine is done by a 2 hydrogen bond, and the pairing between guanine and cytosine is done by 3 hydrogen bonds. This pairing is universal, hence the base pairing of the two strands of the DNA are complementary to each other. Now, out of the two anti-parallel strands, that is this strand and this strand, let's take in this form: one running in 5 3 and other in 5, one running this side and another running this side. So out of the two anti-parallel strand, only one strand carries the genetic information, and it acts as a template for transcription of mRNA. So the strand which carries the genetic information, so out of these two strands, only one is carrying the genetic informations, and the other is just the complementary strand of this strand which is carrying the genetic information. So the strand of the DNA which carries or store genetic information is called as the template strand or the antisense strand, template or antisense strand. So usually this template strand is in 5-3 polarity. So the 5-3 direction nucleotide strand carries the genetic information. So if this side is carrying the genetic information, this side is just the complementary strand. This side do not carry any genetic information, but it carries the base sequence that will bind with the template strands that is carrying the genetic information. So the strand that is carrying the genetic information is called as the template or antisense strand, and the strands which is forming just the complementary strand for this template strand or antisense strand is called as the coding or sense strand. Now the segment of the DNA, let's say these are the two DNA, these are the two strands of the DNA. So the segment of the DNA strand that has codons, when we say codons, we are talking about 3 nitrogenous base, triplet nitrogenous base or 3 nitrogenous base which code for a particular protein is called as the codon. So Let's take an example. Nitrogenous base U, and G. These are the three nitrogenous base. This code for a amino acid called as the leucine. So these three nitrogenous base which is coding for a these three nitrogenous base which is coding for amino acid leucine is called as codon. So codon is referring to triplet nitrogenous base which code for a particular amino acid. So the segment of the DNA that has codons for the synthesis of one polypeptide chain is called as cistrons, and along the length of these cistrons the message coded segments is called as the exon. Now let's say this is the message coded segments. This message coded segment in the length of the DNA is called as exon, and the intervening, so the segment between these two exon, this strand, this segment, the intervening segments between this exon is called as the introns. Now when we say introns, we are referring to the intervening non-coding segments of the DNA. The coding or the message coded segment are called as the exon, and the intervening non-coding segment or the non-message coding segments of the DNA are called as the introns. Now those genes that have both exon and introns along its length is called as the split or interrupted genes. So this is all about the model of the DNA, that is Watson and Crick model.